Good morning, students. In the last period, we completed our first chapter, crop production and management. Today, I am going to start your second chapter, microorganisms, friend and foe. In this chapter, we will learn about meaning of microorganism, which microorganisms are useful for us and which are harmful for us, several methods of food preservation, nitrogen fixation, and nitrogen cycle. So, we will start the chapter. Here, name of the chapter is microorganisms, friend and foe. Friend and foe. Foe means enemy, which are harmful. Now here, in our daily life, in our life, we will saw many more large animals, many more small animals. Like large animals like elephant, blue whale. These are the large animals, and like small insects and ants. These are the small insects, but there are some another small insects are there which cannot be seen with our open eyes, and those are called as microorganisms. We will see the chapter. You have seen several kinds of plants and animals. However, there are the other living organisms around us which we cannot see with eyes alone. These are called microorganisms or microbes. In our daily life, we saw many more plants and many more animals, many more big plants, big animals, many more small plants, small animals. But there are some other organisms are there which cannot be seen with our open eyes, and those are called as a microorganism. For example, you might have observed that during rainy season, moist bread gets spoiled and its surface get covered with the grayish white patches. In 7th standard, we learned this concept already. Observe these patches through a magnifying glass. You will see a tiny black rounded structures. And do you know that these structures are, and where did this come from? We learned this concept in 7th standard. Here, in a rainy season on the moist bread, fungus will be observed which has a grayish white patches, which is called as a fungus, and it is present in the air. When it gets proper condition, proper moisture, proper humidity, that time it will be grows on that food material. Here, our first point, microorganisms. For that, activities are mentioned. Two activities are there. Activity 2.1, collect some moist soil from the field in a beaker and add water to it. After soil particles have settled, after some time, we are aware about this concept also. We will learn in the 6th standard, separation of the substances. In that, we have to take soil, we have to put into the beaker. After that, when we stir, after some time, soil will be settled down. That process is called as a sedimentation decantation process. After the soil have settled, observe a drop of water from the beaker under a microscope, and what do you see? We can see a tiny microorganisms present in that water. Activity 2.2, take a few drops of water from the pond and spread on the glass slide and observe through a microscope. Do you find any tiny organisms moving around? Here, from these two activities, we found that some tiny organisms are present in the soil also, in the water also. From that, we can observe and we can make the observations. These observations show that water and soil are full of tiny organisms. There are many more small organisms are present, though not all of them fall into the category of microbes. From that, all of them, we cannot say that all are microorganisms or microbes. These microorganisms or microbes are small in size, that they cannot be seen with unaided eye. It is already mentioned in the definition of microorganism also. Microorganisms cannot be seen by the open eyes because they are very small in size. Some of these such as fungus that grows on bread can be seen with a magnifying glass, because fungus is grown in a colony. That's why we can see with the open eyes. Others cannot be seen without the help of microorganisms because they are singly developed. They are not developed into the colonies. That is why these are called microorganisms or microbes. Now we will see types of microorganisms. Microorganisms are classified into four major groups. These groups are bacteria, fungus, fungus, protozoa, and some algae. Some algae. Here bacteria, fungus, we are aware about that, protozoa, and some algae. Algae, shaivar. Some of these common microorganisms are shown in figure 2.1 to 2.4. We will see afterwards. Next point, virus. Viruses is also microscopic. Viruses is also microscopic. Now we are aware about that the present situations about COVID-19. The virus are also microscopic. They, however, reproduce only inside the cell of host organism. Inside the cell of host organisms, which may be bacterium or plant or animal. Now we are suffering from this situation. Now we are aware about that these viruses acts on only or reproduce themselves only in the host organisms. Suppose the viruses are spread on that any other material, any other material, on that it cannot be reproduced. But suppose they get the host organisms like plant, other bacterium or animals, they will reproduce themselves. They required host organisms, they required other body to develop themselves, which may be bacterium, plant or animals. Some of the viruses are shown in figure 2.5. Common ailments like cold, ailments like diseases, common diseases like cold, influenza, it means flu. In local language we are saying that tap, and most coughs are caused by viruses. Serious diseases like polio and chickenpox also caused by viruses. Now we are already aware about that viruses, polio and chickenpox, it is caused by the viruses. Diseases like dysentery and malaria caused by protozoa. Diseases like dysentery and malaria caused by protozoa. You read this carefully because in the exam maybe it is asked for match the column, which diseases caused by which virus or bacteria or other other microorganism. Disease like dysentery and malaria caused by protozoa, where the typhoid and tuberculosis are bacterial diseases. You have learned about some of these microorganisms in 6th and 7th standard. We already learned some microorganisms. What is the use of some microorganism in last academic year and 6th standard also, like nitrogen fixation, which organism is required, rhizobium, about rhizobium, lactobacillus. We learned these concepts already in last academic year. Here the diagrams are mentioned here. Here you can see the first diagram, figure 2.1, bacteria. Here two types of bacteria will be there. First one, spiral bacteria. Second one, rod shaped bacteria. Next, here downside, algae. Here Chlamydomonas and Spirogyra. Two types of algae are mentioned here. Third figure 2.3, protozoa, which are single celled organisms like amoeba. We already learned in a 7th standard in chapter 2nd, nutrition in animal. In that we learn about the nutrition, feeding and digestion system in amoeba. Then paramecium. Next one are fungus. Here bread mold, bread वरती आलेली बुरशी, penicillium and aspergillus. These are the types of fungus. Now the last figure is 2.5, figure 2.5, viruses. These are the viruses. Here upper side one diagram is given, upper side in a 2.5. there it is closely matchable with the nowadays coronavirus is closely look like this. And these are the concepts, these are the diagrams of microorganisms. Now where do the microorganisms live? Here now we are from the starting of the chapter we will learn about microorganisms. What is the meaning of microorganisms? Can they see with the open eyes? Types of microorganisms. Then figure. Now where they are living, microorganisms, where they live, we will find out. Microorganisms may be single celled. Like bacteria, some algae and protozoa. protozoa or multicellular such as algae and fungus, such as algae and fungi. Here, microorganisms. There are two types of microorganisms will be there. One, they are single-celled microorganisms, and another as multicellular organisms. Here, single-celled organisms are bacteria, some algae and protozoa, and multicellular such as an algae and fungus. They can see with the open eyes. They can survive under all types of environment. They can survive under the all types of environment, ranging from ice cold climate, ranging from ice cold climate टू हॉट स्प्रिंजेस थंड वातावरणापासून एकदम गरम वातावरणापर्यंत सुद्धा ते राहू शकतात. अँड डेझर्ट टू मार्शलँड, वाळवंटापासून ते दलदलीच्या प्रदेशापर्यंत सुद्धा ते राहू शकतात. दे आर ऑल्सो फाउंड इनसाइड द बॉडीज ऑफ ॲनिमल्स इन्क्लुडिंग ह्युमन. in 7th standard in second chapter we learn this. Some microorganisms or some bacteria will be available, will be present in the human body also and animal body also. Some microorganisms grow on other organisms while other exist freely, or other exist freely. Microorganisms like amoeba can live alone while fungus and bacteria may live in a colony. Some microorganisms like amoeba, we already learned this concept. Amoeba live alone, but fungus and bacteria lives in a colonies. Now the next point, microorganism and us. Now, what is the relation between the microorganisms with the human beings and with the other animals? What is the relation between both of us, microorganisms and us? Already in the chapter name it is mentioned microorganisms, friend and foe. From that we can understand that some microorganisms are useful for us and some microorganisms are harmful for us. We will see. Microorganisms play an important role in our lives. Some of them are beneficial in many ways, whereas the other are harmful and causes diseases. Let us study about them in detail. In this chapter, the main concept is that we have to learn about the microorganisms, how they are useful for us and how they are harmful for us. Here, friendly microorganisms. Microorganisms are used for various purposes. They are used in preparation of curd, bread and cake. Those are all are bakery products. There it is used. Microorganisms have been used the production of alcohol since ages. खूप वर्षापासून मायक्रोऑर्गॅनिझम्स काय करतात, अल्कोहोल तयार करण्यासाठी वापरले जातात. That also we will see afterwards. Here, see, they are also used in cleaning up the environment. In last 7th standard in the forest chapter, forest our lifeline, in that we will learn this concept. We already learned this concept, that cleaning up our environment. Some bacteria are present in the soil, those are helpful for us to cleaning up our environment. They are also used in cleaning up our environment. For example, the organic waste like vegetable peels, remains of animals, feces, etc. are broken down into the harmless and usable substance by bacteria. In a forest chapter we learned that in forest humus is produced naturally. Here, suppose any plant or any animal will be dying, which will stay on the upper side of the soil. From that, after some days it will be vanished by the microorganisms. Microorganisms convert them into the humus, and which is useful for us also and plants also, because plants they use this again and they get the nutrients from that. Recall the bacteria are also used in the preparation of medicines. In agriculture, they are used in increased soil fertility by fixing nitrogen. The rhizobium bacteria, we are already aware about the rhizobium bacteria, which is used for nitrogen fixation. Next one, making curd and bread. You have learned in class 7 that milk is turned into the curd by bacteria. bacteria. We learn this concept in a 7th standard, making curd and bread. Here in that we have learned in a 7th standard, milk is turned into the curd by the bacteria. Curd contains several microorganisms. Of these, the bacterium lactobacillus, which is present in the curd, that promotes the formation of curd. It multiplies in the milk and convert into the curd. Bacteria also involved in this making of cheese, pickles, and many other food items. An important ingredient of rava, idlis, and baturas is curd. And can you guess why? Here, how we are making curd from the milk. Here we are aware about that we have to put some curd material in the milk. In the curd, lactobacillus is present, which promotes the formation of the curd, and it multiplies itself in the milk and converted into the curd. At the last, one question is asked: why curd is important ingredient in idlis and bhaturas? Why curd is important ingredient in idlis and bhaturas? I will tell you the answer. Curd contain lactobacillus bacteria, which is mentioned upper side. In the curd, lactobacillus bacteria will be there, which undergoes anaerobic respiration. Here in the 7th standard, respiration in organisms, in that we learn about the aerobic respiration and anaerobic respiration. The curd contains lactobacillus bacteria, which undergoes in a anaerobic respiration, and this process it acts on a sugar of the flour and release carbon dioxide, and this gas makes dough rise, and when baked or fried, that time this gas escapes and leaving the idlis and bhaturas soft and spongy. To make idlis soft and spongy, the curd is most important, because there lactobacillus bacteria will be present. When we fried or when we baked that food item, that time what will happen? This gas escapes from that food material, and it makes idlis soft and spongy. Here one activity is mentioned, activity 2.3. Take half kg of flour, it means atta or maida, we can take, add some sugar and mix with water, warm water, add a small amount of yeast powder, and knead it to make the soft dough. We have to take half kg of flour, atta or maida we can take, we have to add some sugar and mix with the warm water, and add a small amount of yeast powder, which is available in bakeries also, and knead it, make soft dough. मळायचे ते पीठ मळणे. And what do you observe after 2 hours? After 2 hours we will find that the dough will be rise. Then how it is possible? Yeast reproduces rapidly and produces carbon dioxide, as we learn in the lactobacillus also, in the concept of the lactobacillus also. In the same way that yeast produces rapidly and produces carbon dioxide during the respiration, that's why the bubble of the gases fill the dough and increase its volume, and this is the basis of the use of yeast in a bakery industry for making breads, pastries, and cakes. Here, why the yeast is used in the bakery products? The main concept, what is the main concept behind that? The yeast produces rapidly and produces carbon dioxide during the respiration, and the bubble of gases increases the volume of the dough, and that's why they are using in the bakery products. Today up to this we will cover, remaining part we will see in the next period.